Hello, my name is Nicolas Corradi. I'm the principal investigator of my laboratory located in the Department of Biology at the University of Ottawa. Over the past 8 years, my lab has hosted and trained over 70 different personnel. Lab training includes postdoctoral fellows, as well as PhD, master and honor students. Undergrad and visiting international students and researchers are also an important part of my team. Presently, my lab is composed of 1 postdoctoral fellow, 3 grad students, as well as 2 honor students and several volunteers and work-study students. My laboratory uses tools of molecular biology and bioinformatics to better understand the biology of microorganisms that are important from a medical perspective and from an ecological perspective. One of the organisms that we study are called arbuscular mycorrhizal fungi. These fungi facilitate the capture of soil nutrients by plants by extending small root-like structure called hyphae. These hyphae penetrate the roots of plants, resulting in a complex nutrients exchange network between the plants and the fungus. Generally, in the presence of these fungi, plants grow much better. Ecosystems are also much more diverse, and plants are better protected against pathogens. There are 2 main ways you can use to better understand these fungi. One, you can look under a microscope for hours, hoping to discover something exciting. This approach, however, is very time-consuming and often results in frustration, as these tiny organisms do not always like to cooperate with us. Another, perhaps more efficient way to look at their biology and better understand their way of living is to look at their DNA. Specifically, by sequencing their DNA, we can identify the genes and proteins that they use to survive, adapt, and propagate. To do so, we cultivate this organism in the lab, extract their DNA and RNA, and analyze their content using tools of molecular biology and bioinformatics.
Hi, my name is Gökalp and I'm a PhD student from Turkey. My research aims to understand the DNA content of this organism using a combination of molecular and bioinformatics techniques.
My name is Stephanie and I study the interactions between the strains and varieties of these fungi using a combination of microscopy and molecular biology techniques in order to better understand the biology of these microbial organisms.
Hello, my name is Matthew and I study the RNA of this fungus to understand which genes are being expressed during different time points of their life cycle. Overall, our research comes together to better understand how these microbes create genetic diversity. This is important in order to understand the role this fungus plays in helping plants and crops grow better and to produce a healthier and more diverse ecosystem.
Hi, I'm Madhu, postdoc from India. I have expertise in bioinformatics. Using the tools of bioinformatics, I can identify the genetic components of the beneficial fungi that helps the plants to grow better.
The fungal organisms that we work on are essential for our everyday lives and play a key role in the growth and health of most plant species. They are also often used by industry, and we closely collaborate with industry partners that specialize in organic agricultural application, as we try to produce fungal strains that are better suited for certain plants, for example, or certain specific conditions. Ultimately, this will help the green economy and the environment by reducing the overall use of polluting fertilizers.